Hello, and welcome to this podcast for Epigenetics. In this podcast, what I'd like to do is to revisit some of the concepts from genetics. So let's start with a basic definition of genetics. It's essentially the study of heredity. And also the variation of inherited traits. Next, let's think about our good friend, the Central Dogma. And essentially, the Central Dogma says that you start with DNA, it moves to RNA, and then it moves to protein. Now, we know there are a lot of flaws to this. Not all pieces of DNA will actually be turned into RNA or protein. Some have regulatory functions. Some only make RNA, and the RNA is the functional unit and doesn't make protein. So this is just a general idea, but it's a good place to start. And I'm going to go over some features of this, but I'm also going to remind you of some terms and definitions as we go through this. So first, remember, we're starting with DNA, and DNA has this double helical shape here. But not all of this is turned into RNA, only small regions. These regions which we call genes. And this gene here is transcribed into a messenger RNA molecule in this example. And let's draw the messenger RNA like this with the G cap and a short poly A tail. But this is the RNA. And then the RNA itself is translated, which would go on this arrow, and transcribed would go on this arrow. It's translated into a protein. And a folio full of protein looks usually like some kind of blob here. Now let's give this gene here a specific name that we're talking about here. And let's call it GLUT2. The messenger RNA it makes is a GLUT2 messenger RNA. And over here, this is our GLUT2 protein. We would think of GLUT2 here as this organism genotype. In this case, what I'm drawing here is a non-mutated GLUT2. And so this is the genotype for this individual. The genotype is just the genetic makeup of that individual. Then over here, the protein, we call this the phenotype. Phenotypes are just the observable traits that are generated from the genotype. Sometimes, as I mentioned earlier, the phenotype might just be the RNA that's produced. But in this case, we're saying the phenotype is associated with this protein. I also want to just put in parentheses here trait, because sometimes I'll interchange these two terms, phenotype and trait. Probably shouldn't, but I do. So what is GLUT2 doing? So let's draw a cell here. And what GLUT2 does, its phenotype or its trait, is that it makes this receptor. And this receptor allows glucose to enter into the cell. Now, we know that within a population of individuals, there is some genetic variation. And that is because not everybody will have this exact same gene, or rather the exact same sequence of this gene. Sometimes there are different alleles. So let's, down here, just define allele. And then I'm going to erase it because I need this room to draw. And these are just alternate forms of a gene that arise due to mutations. Now, this being an epigenetics class, we're not going to focus a lot on mutations, but it's good for us to think about different alleles within a population. So let's, for the sake of this discussion here, let's draw a different allele of GLUT2. So this is a different individual. They also have this gene called GLUT2. But this time, there is a mutation in it. Now, some mutations have a benefit, some have a negative consequence. But in this case, this mutation, but all mutations change the sequence of a gene. So it will still make this messenger RNA, but this messenger RNA now will be carrying forward that mutation that's found in the gene. It will make a protein, but there's a slight difference in the way it folds because of this mutation. And so we'll draw our cell here, and it still makes this receptor. However, it folds a little differently, as I've drawn here. And in this case, it still takes in glucose, but less efficiently, which I'll indicate with this broken arrow or dotted line. So in both cases, we have a genotype and a phenotype. You go from a gene to an RNA to a protein. The difference, though, is due to genetic variation, we have different alleles. And this allele here, in this bottom one, results in an altered phenotype. And in this case, will lead to a metabolism disorder. Now, before I move on to the next slide, I want to remind us that our knowledge of what's shown on this picture in a very simplified form is the result of many decades worth of research understanding and breaking down this central dogma and thinking about mutations. Where we're going to spend a lot of our time this semester, though, in epigenetics is not talking about mutations, but talking about how we can have these altered phenotypes, not due to mutations, but due to how this gene, this GLUT2 gene, is regulated. So you could have a GLUT2 wild type gene here, non-mutated, but there is some sort of alteration into the way it is regulated that can lead to a non-functional or a less functional protein as an end result. Next, what I'd like to do is just spend a little bit of time talking about model organisms. Model organisms are incredibly important in the study of genetics. And as we'll learn, they're also incredibly important in the study of epigenetics. And so let's just spend a little bit of time listing some characteristics of model organisms. Similar characteristics that you've probably written in several other classes. So this is maybe just a review for some of you. They will typically mature rapidly. You don't have to wait years for them to mature. They might mature in a matter of hours or days, or maybe weeks in some cases. Related to maturing rapidly, they have a rapid generation time. Model organisms are easily manipulated in a lab. Model organisms produce large amounts of progeny. All right, now that we've talked about some common features found amongst model organisms, I'd like to make a list of some examples of model organisms. And let's start off with E. coli, an example of a bacteria. This is a very valuable model organism. It's the workhorse amongst bacteria. In addition to genetic studies, it is also used with some epigenetic studies as well. Still thinking about single-celled organisms, but now we're going to talk about a single-cell organism that's a eukaryote, and this would be Saccharomyces cerevisiae, which is a yeast, which of course is an early eukaryote. Now if we want to start thinking about some multicellular eukaryotes, two very common ones are C. elegans and Drosophila melanogaster. These have some advantages over Saccharomyces in that they are multicellular. They also have a fairly detailed developmental pathway. A disadvantage might be that they have a shorter generation time. As we'll see, every model organism has its advantages and disadvantages, and it's just best to pick the model organism that is best suited to the research question you're asking. Another multicellular eukaryote that's commonly used is the zebrafish. Now, a common model organism that is a plant would be Arabidopsis thaliana. Some other model organisms that are often used but typically have a longer generation time, so are not always ideal for genetic studies, but they're still model organisms, would be Mus musculus, or the common mouse, Xenopus, frog, and the last one I'm going to list, it's not really a model organism, but it's a common way that we study cells that come from humans, and that would be, I'm just going to indicate it down here as other, and that would be cell culture. Not exactly a model organism in the way we think of model organisms, but certainly a very valuable way that we study cells, particularly human cells, because humans make really lousy model organisms. So here's a pretty good list. Again, there are many others, so I'm not trying to slight anyone's favorite model organism here, but this is a good list. And what we'll find is not only have these proven to be very valuable in the study of genetics, they have also proven to be very valuable in the study of epigenetics. And so we'll encounter several of these as we move forward this semester. Well, that's all I have for this video podcast. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye for now.